talk in such a great event. I'm Gil-tae at Busan University. So I'm going to talk about some ongoing research topics, including identification of gene disease associations. Before starting my talk, and I want to share some couple of pictures. The left one is like when I joined Mike Chang's lab as a postdoc, who is the keynote speaker today. And so he was many years ago when we attended Eastern genetics meeting in Frankfurt, Germany. And so I worked there for East Genome database to assemble East genome sequences and its annotations and so on. Yeah, so it was a great time. I spent 4 years there. And yeah, this is another picture which means to me a lot. And so that was taken last year. So since I joined Busan University, so I allowed to have a visit, allowed some of my students to visit the laboratory. So I guess we had 2 or 3 students to visit every year. And I counted the total of students, it was like 15 so far. And so I think it was a great experience. And so then we could continue our collaborations since I left his lab. And so my group, originally I studied genomics and I analyzed the genomics data. And now my laboratory is working on integrating the genomics various genomics data and other types of data from hospitals, such as electronic health records. And so we want to apply, we are applying machine learning artificial intelligence for analyzing those data. And why do we care about this? Because we want to, as Professor Tae mentioned in his talk, and so we want to realize, we are aiming at realizing precision medicine. So based on people's genetics information, we want to apply different personalized diagnosis treatments so that all patients can have a positive effect. And so this one could have one big milestone of the completion of the human genome project. So the human reference sequence was completed in 2003. Actually, East genome was, as Mark mentioned, East genome is the first organism of which the sequence, the whole genome sequence was completed here. Yes. So that's the time that the genome database was initiated and created. Yes, a good milestone. And then, since the genome sequence, human genome sequence was completed, and the encoded consortium was initiated, investigated for more than 20 years. I guess that these numbers could be out of the date, but I got from encoded data, encoded DCC, and so a lot of research groups have participated in this encoded project. So the aim of the project to understand the function of the genes and discovering additional functional elements using various types of experimental data, such as ChIP-seq, RNA-seq, a bunch of experimental data. And so more than 300 software tools have been developed, and some use machine learning. And so the goal, finally, the aim of the project, I guess, we want to discover important biomarkers associated with diseases. And so, so my research, my group focuses in this theme. So we discover biomarkers associated diseases. And the first, the basic step to this is detection of genomic variation. So this is collaborative work. So my group just helped the projects led by Alex Urban at Stanford, and so this happened. This collaboration happened when visited Stanford in 2017, something like that, 2018. And so he worked on, Alex Urban's group worked on human cell, cancer cell line genomes. And so this is the cancer genomes. There are a lot of structural variations happening. And so we help to analyze determining copy variation and analyzing them. And so one popular cell line is K563, leukemia cell line, and another is HER2, liver cancer cell line. In addition, once we figure out genomic variation, then we want to verify, validate their function of the variation using some other model organisms, by additional experiments, and so that's why these kind of studies are done in non-human species, such as yeast, mouse, something like that. And so this one is a continuous collaboration with Alex Urban. So they sequenced marmoset genomes, and so they raised the question. So they wanted to determine genomic variation of their marmoset individual genomes. And there is one popular tool called the Git variant that was developed by Google DeepMind. So there were a lot of software tools to detect genome variation, like SNPs, single nucleotide polymorphism, or small intels. But with Google DeepMind, they applied deep learning, and so then this tool has been dominant for determining human genome variation. But this tool needs some prerequisite information from well-known variants which have been already verified by population genomics studies. So that kind of information is enriched in humans, but not in other non-human species like mice. So for other non-human species, we want to use a deep variant. We don't have this. So then we had a lot of false positive research. So we developed to refine the outcome of deep variant to filter out a lot of false positives using some machine learning approaches. So our pipeline improved a lot. So we applied this pipeline to determine false positive codes for rhesus genomes, which we studied in 2020, and so they had quite good quality assemblies. So it was already verified, so we could measure the performance of our pipeline, and so we Yeah, so this work was done by Jung Choi, who's a PhD student in my laboratory, and so we got this. We was selected as a top in ISND last year, and so we just submitted this pipeline paper to a journal. Yeah. So it's under review. So once we figure out genomic variations, then so we want to link like some genomic variants to species we are interested in. To do this, like understand the function of the genomic variation, or like genes are important, and so other types of epigenetic experiments are done, and so ChIP-seq is one of the common experiments to find kind of transcription binding sites, understanding the histomodification, and there were kind of popular pipelines to determine to analyze the ChIP-seq data, and so ChIP-seq we have this kind of data, like so all kinds of reads are mapped to the reference genome, and so then some reads are mapped to that, and so we call like some this kind of signal as peaks, and so but in cancer genomes, like it's there are some different kinds of patterns. So what happens in the people, and so when they ran the pipeline for calling peaks for ChIP-seq, and then they have to examine, like they have to examine the codes are correct or not. So sometimes they use the visualization tool, and so at that time, but this is quite hard to examine all the bits called by the pipeline for the whole genome. So we thought, if experts mark some labels or whether the peaks called true or false positive, and so we use those labeled data to train a model to pick out false positives. And so we designed a CNN model optimized for this data. So this data is actually signals into one-dimensional. So our pipeline actually shows the highest F1 score, and so this one was published in 2020. And this one was done by O, who was a master's degree student in my laboratory, who visited my lecture's lab. And this project was initiated and we visited his lab. And so these kind of experiments are quite expensive, and so but one time, like PI at the University Hospital, talked to me, like she wants to do this kind of experiments, like genetics experiments, like ChIP-seq, RNA-seq, But she asked me, she doesn't have enough money to do like whole genome wide or high throughput sequencing or something like that. And so she wants to know like what kind of genes she needs to focus on. So and yeah, she has a specific disease of her interest. So we had to figure out to help her, and I realized that that's the whole actually study right we are doing, and so it's not an easy problem, and researched like research articles, and there's a database called DGNet that was actually created late 2000, maybe 18, 17, and yeah, so I think they curate the data from articles, research articles published, and they have some known association of genes versus diseases, and they put to that information in DGNet database. So we wanted to try this database, but part of the disease she is interested in is not quite common, so that can be quite studied well in this, especially in genetics field. So we had a problem, and so this is a matrix, and when some associations are known, we have some scores or something like that. But mostly, as you see, mostly are empty. Yeah, we don't know. They are not known yet. And and we raised the question how to fill in those empty spots, and one of my students told me he wanted to apply a recommendation system which was used for Netflix recommendation. Okay, yeah. So in terms of problem, it's the same, right? In Netflix, we have users and movies. In our case, genes versus diseases, right? And there are empty ratings, right? Hasn't been rated yet, and so so we wanted to apply it, but actually it didn't work out well because the association between genes and diseases are more complex, much more complex than that system. So this kind of additional information underneath the association of genes and diseases. So what kind of approach could work well? And so nowadays, graph-based representation learning is actually suggested a lot, proposed a lot. So my group was decided to apply this approach. So the relationships, interactions of genes and diseases can be formulated as a graph in data structure, and so then they are embedded into low dimensional vectors, and then these graph representations can be fed into downstream tasks like neuron architectures to identify node class or link prediction. So when we took a look at the gene disease association problem, actually the relationships are happening among heterogeneous nodes, like genes, disease, and in addition, we need to take care of other kinds of data resources which are related to gene disease associations. Maybe for genes, we need more information about gene ontology, or disease ontology, or some human phenotype ontology, and so on. There are lot of genomic resources. So we designed to incorporate those kind of information and to build kind of heterogeneous graphs, and so and we applied the graph convolutional neural network, and we named it HAGCN, and so and we use the known gene disease associations from DCNet to these graphs, and so we measured the performance of our method, and so these are like graph-based networks for the gene disease association problem, and our pipeline shows the best performance score, and so this is about to submit a paper. I think the manuscript is ready to submit, and so this work was done by Kim Kim in my laboratory. Yeah, so this is how to, oh, so this is different. How to figure out unknown disease gene associations, and in addition to genetics side, for precision medicine, I think it's important to understand patients' data, and so we had a collaboration with the clinical expert, PI, in cardiology at Busan National University Hospital. Actually, who is my wife actually. Yeah, so we always talk about collaboration between us, and so she suggested some well-known data for the Korean acute myocardial infarction registry, from registry, and so it's called CAMIL. So we use this CAMIL data. So that was collected by the nationwide registry, which 53 centers have been participated. Actually, during 3 years, but it's still expanding. More data added to this registry, and so in total we have 15,000 patients, and each patient had like 614 features, and so there is some information about mortality. Some hospital mortality, 6-month mortality, and 12-month mortality, and so we tried to build a model to estimate the patient's mortality using these data. And so we had to apply some pre-processing steps, and finally we developed a pipeline called RIAS, which is Reliable Interpreter AI System, and this is about like, so we take data from CAMIL, and we apply 3 different models, like based on XGBoost, multilayer perceptron, and FFT transformer, and for hyperparameters were optimized using QNA, and we use the shape value for global and local explanation, so that we can figure out what features are important to predict the mortality of my patients. And normally in this kind of classification problem, the output of the model is a kind of a score between 0 and 1, and so it could be a probability, maybe the probability, but actually it's not right score, that it doesn't match actually the likelihood. So we calibrated this score to more accurately represent the true practice likelihood. Yeah. And we provided counterfactual explanations using DICE. This means so because we determine what kind of features are important to predict mortality, the risk of mortality of AMI patient, and so we made kind of what if scenarios. So we asked like what if the kind of feature is corrected, modified to different like range or different option maybe. So actually there was a estimate to my right. What if this one was changed? So that some features could be modified, sometimes like such like dosage medicine, right? Yeah. But maybe so we just ask the question about the features, the correction of features which can be modified. Yeah. So this is the performance of our model for predicting mortality, and this is an example of some features, important features to predict mortality. And this is the example of counter example. We asked to the clinical expert again. So the figure above is like originally in the data, like the people, the patients didn't have beta blocker prescription, and so but there was an important feature of the patient which was determined by the RIAS system, the high mortality. And so we requested to change it to have the patient beta blocker prescription, and then the confidence score dropped significantly from 95% to 38%. So this is quite surprising, and so and this work was it's gonna be up here in Journal of the American Medical Genomics Association next month. Yeah. This work was done by Kim, who is an investigator in my laboratory. Yeah, so this is what my laboratory is studying so far, and so like various healthcare big data such as genomics data and hospital patients data from hospitals, and like some high computing technologies and devices could finally lead us to realize the precision medicine. Yes. And so I give thanks to all hard work to all my students and great collaboration with Mike Cherry and Alex and thank you for your attention.